Hi, my name is James Nicodemus, and my presentation is going to be on cystic fibrosis. And we'll talk about what exactly is cystic fibrosis. And what it is a defect in a gene known as the cystic fibrosis transmembrane conductance regulator, and it's known as the CFTR. And this particular gene is located on one of the autosomal chromosomes, number 7, at position 31.2. So it's at the bottom half of this gene right there. And it is responsible for translating this particular protein known as the CFTR protein. And this shows an example of it here, of the protein itself sitting in and through a cell membrane, the phospholipid bilayer. And this is an ion channel protein, specifically for chloride ions. And so this is responsible for either letting chloride ions out or absorbing them back into the cell. And when there's a defect in this, that can cause some problems. Here is the amino acid sequence for this particular protein, and it's quite a long one here. It's 1480 amino acids long. And it's kind of the tertiary structure of it, as you saw on the previous slide, is kind of creates a channel that goes through the cell. Here's an example of the CFTR proteins in a normal operation, sitting on the cell membrane and through it there. So they're allowing the chloride ions in and out of the cell. Subsequently, when you have the chloride ions, you also have the sodium ions. They tend to work together. So if you don't have one, then you typically don't have the other. So let's talk about what happens in cystic fibrosis itself. This probably the main one that you'll see a lot is the thick mucus in the lungs, and this is pretty typical with cystic fibrosis. They have a lot of respiratory problems. You see this in young kids. They have difficulty breathing. They get sick a lot with pneumonia, bacterial infections. Digestive problems are a big deal too in these young children. Typically with things not working right in the digestive tract, as well as the pancreas there, a lot of these kids don't gain a lot of weight. They have some difficulty with their digestive problems. With the pancreas, you can see damage to the pancreas, diabetes type things, some of the different pancreatic enzymes. And particularly with males, you'll see some problems with the reproductive problems where they typically, a lot of the males do not tend to have, tend to either not have a vas difference, or it's tends to be plugged with thick mucus. And the mucus is a big problem with really all of these here, and we'll go into a little more detail of why that is. If we look at a normal CFTR protein here, the let's say this is the lining of one of the epithelial cells of the lungs. Normally you'll have little cilia there on the outside of that. You'll have a mucus that is produced, and then the cells will pump some of this chloride ions, subsequently the sodium, onto the surface of the cells, which attracts water and thins the mucus. So you normally would have a nice thin layer of mucus that with the little cilia that push along the mucus, and it helps clear out different debris and bacteria and impurities that enter the lungs, and it helps part of the cleaning process. With a defect in the CFTR protein, the chloride ions are not allowed out off to the surface, which subsequently the water does not follow that, and this mucus becomes thick. The cilia get flattened out, and they're not able to move that mucus along. You can kind of see here that mucus builds up. Sometimes it gets stuck in there. The bacteria begin to infiltrate. You get inflammation. Sometimes they'll have like mucus plugs, and bacterial pneumonia is a big problem with the cystic fibrosis. You also see this in the digestive tract, as well as the vast difference of males and the pancreas as well. So let's talk about the inheritance and the prevalence of cystic fibrosis. It is an autosomal recessive condition, which means that it does take two copies of the, we'll call this the small a, or the recessive allele, to pass along the condition. And the prevalence of this you'll see in, these are statistics for North America, but about 1 in 2,500 to 3,500 of the Caucasians. You'll see it less in the African American and Asian babies. The history goes back a little bit. I found an interesting quote from the Middle Ages, and this was one that they said, "Woe is the child who tastes salty from a kiss on the brow, for he is cursed and soon must die." So you would wonder, why would one of these kids have extra salt on their skin? And this is exactly what happens when we produce sweat. We will normally produce a salty with water and sodium and chloride from the sweat, and then as the sweat begins to go through the duct to the surface, there are some of these CFTR proteins that help reabsorb the ions back into the cell. Without that, the salt becomes extremely salty and then ends up on the skin surface. Subsequently, this is one of the ways that they test for cystic fibrosis, by doing what they call a salt test, and they can measure the sodium and chloride content on the surface of the skin. But as far as the history goes on that, it goes back quite a ways. This first quote was from the Middle Ages. The first doctor, Dorothy Anderson, described this as cystic fibrosis of the pancreas. This is where they found that when they were doing post-op or post-mortem exams, they would find that there was damage and thick mucus built up in the pancreas. They hadn't really figured it out as far as the lungs go. Later on, they described it as mucoviscidosis, and this was the thick mucus. That's how they described that in the 1980s, and then they did describe this, that there was a protein defect that was for the order, and then in 1989, this was the particular gene that was identified. When we talk about how this can be passed along here, there's really six examples of combinations of parents that can produce offspring. In the first example here, we have, big A is the normal allele, and small a would be the cystic fibrosis defect. So obviously, if you have neither parent is a carrier and they have a normal alleles there, big A and big A, 100% of their offspring are going to be the big A variety. If you have a carrier, which would be a big A, little a, and a normal allele, big A, then you have a 50% chance of producing a carrier as an offspring. The normal, or one of the big ones you see, is where both parents are carriers, and this would be big A, little a, big A, little a, and this would be your offspring chance. You would have big A, you would have or a 25% chance of a non-carrier. You'd have a 50% chance of a carrier, and you'd have a 25% chance of child being born with the cystic fibrosis. If you have one parent that has cystic fibrosis and one parent that does not, then you have a 50% chance of, or not 50, you have a 100% chance of all children being born as carriers. And then if you have a cystic fibrosis parent and a carrier, then you have a 50% chance of either having a child with cystic fibrosis or 50% chance with being a carrier. And then obviously, if both parents have cystic fibrosis, there's a 100% chance of all kids having the disease. This is just an idea of a pedigree here. If you can see that this family started off with parents that were both carriers, then you would have quite a variety. You see some that have that are non-carriers, some that are carriers, and then two here that actually have the disease. And this will kind of go through and show you just kind of how, with a lot of pedigrees, you'll see some things skip a generation, not show up in every single generation here. So with this is a pretty typical pedigree chart of what you would see with cystic fibrosis. There have been about 1,700 identified mutations with CFTR, and one of the more common ones Normally you would see CFTR. This is a normal situation here where it's the CFTR protein is made, doing its job here. One of the more common ones, you have deletions or mutations or frame shifts there. You'll see where there's just no functional CFTR made. Some are misfolded; they do not make it to the cell surface. Some they don't function properly. Some the channel is faulty; it doesn't let enough ions out, or there is such a small quantity of them made that it's doesn't do the right job. So those are some of the different mutations that can happen. Typically for genetic testing, most I looked up here with the ARUP, which does a variety of testings. They will look at many different things. You can look for fetal screenings. You can look. One of the more common ones is where you'll see two parents go in to see if either one of them are carriers, and then what their chances are of becoming what their chances of their offspring having the disease or being a carrier. A lot of times they'll go through, once they see different with the children, they'll see some of the different symptoms showing up. So they'll have this one here is much of what they do at ARUP. There is they used to be like 32 variants. Now they're looking at like 165 there. So this is a very, very detailed screening for this, and they can look at whether or not some of these children are showing some of the more common or less common mutations for the disease. What they found out, and for cystic fibrosis, this is part of the typical newborn screening as well. Because what they like to find out is if they can identify this early, then there's a lot of things that can there's a lot of things that they can do to help add years to their life, better nutrition, and lung function if they can catch us at an early age here. And then I'll go ahead and move that a little bit so you can see all the references. And thank you so much for watching this presentation.